Let's move on to some new helminths. This group of helminths is called Trematodes, which are leaf-shaped flatworms, also known as flukes. The life cycle includes a snail intermediate host. Trematodes have specialized organs for reproduction and attachment. Like cestodes, they are also hermaphroditic, except for a few species, such as the schistosomes. In this lecture, we'll look at the liver fluke, Fasciola hepatica, and the blood fluke, Schistosoma. The sheep liver fluke is mainly an infection of the bile ducts and liver. In the early or acute phase, gastrointestinal problems result from the parasite's migration from the intestines to and through the liver, causing nausea, vomiting, and abdominal pain or tenderness. In the chronic phase, after parasites settle in the bile ducts, inflammation and blockage of the bile ducts, liver, gallbladder, and pancreas can occur. This is a zoonotic disease that results from ingesting freshwater plants containing encysted larvae associated especially with the consumption of raw watercress and undercooked livers of infected sheep. The reservoir hosts are sheep and cattle. Although sheep liver fluke infections occur worldwide, it is most frequently found in sheep and cattle rearing countries in many parts of the world, although it is not as prevalent in North America. The adult stage of the liver fluke infects the liver and produces eggs that are passed in fecal matter to the environment. Ciliated larvae hatch, then penetrate the snail intermediate host. They undergo several developmental stages in the snail and are released as free-swimming cercariae, which encyst as metacercariae on water plants, such as watercress. When sheep and humans eat raw, contaminated aquatic vegetation, they become infected. The metacercariae exist in the duodenum and migrate to biliary ducts in the liver, where they mature into adults and continue the life cycle. Adult liver flukes are large enough that they do not need magnification to be observed or identified. They're 3 centimeters long and 1.5 centimeters wide and leaf-shaped. You can see a liver fluke in the picture on the right at the top. The eggs are oval-shaped and are slightly larger in length than 1/10 of a millimeter. So, a microscope is still required to view them, and you can see one in the picture at the bottom. Liver fluke infections can be confirmed by the detection and identification of eggs in fecal specimens. Usually more than one is required. As well, duodenum and biliary aspirates can be used. Alternatively, serology may also be used. Treatment with anti-helminthics is available. Transmission can be prevented by thorough cooking of vegetables that are grown in fields irrigated with polluted water and avoiding the consumption of raw watercress and other aquatic plants from these areas. As well, organs from potentially infected animals, such as liver, should be cooked adequately, even though they may become less palatable. Strict control of growth and sale of watercress and other edible water plants should be implemented. The next type of flukes we'll look at are the blood flukes, which are very different from intestinal flukes. The ones we'll cover in today's lecture are schistosomes. They cause schistosomiasis, which is also known as bilharzia. This disease also has 2 phases. The initial phase presents with a rash or itchy skin a few days after infection. Then fever, chills, cough, and muscle aches a few months post-infection. The chronic phase, which may persist for years if untreated, results in abdominal pain, enlarged livers, blood in the stool or blood in the urine, and problems passing urine. There is also an increased risk of bladder cancer. Schistosomiasis is transmitted through exposure to water that contains infectious cercariae, and swimming and bathing are some examples of high-risk activities. It is second only to malaria as the most devastating parasitic disease in the world, with 200 million infections worldwide. There are 3 major species that are pathogenic to humans: Schistosoma mansoni, which is found in Africa, South America, and the Caribbean; Schistosoma hematobium, which is found in Africa and the Middle East; and Schistosoma japonicum, which is found in Asia. This disease is not endemic in North America. Infected humans pass eggs in their feces or urine into the environment. Ciliated larvae hatch, then penetrate the snail intermediate host. They undergo several developmental stages in the snail and are released into an aquatic environment as free-swimming cercariae. When these free-swimming infective cercariae penetrate skin, they lose their tail to become schistosomular, which migrate to the veins in the intestines or bladder depending on their species. They mature into adult worms, and the females lay their eggs in the venules, which pass through tissue to the intestines or bladder, and then pass out of the body either in feces or urine. Both Schistosoma mansoni and Schistosoma japonicum are found in the intestines, whereas Schistosoma hematobium is found in the bladder and ureters. Adult worms can be either male or female and are cylindrical in shape, unlike most trematodes. The male is short and stumpy, around 1½ centimeters long, and has a copulatory groove, whereas the female is long and slender, around twice the length of the male worm. In the picture, you can see a pair of mating Schistosoma mansoni trematodes. Note that the thinner female is cradled inside the thicker male worm's gonocophoral canal or copulatory groove. The eggs have spines and are between 1/10 to 2/10 of a millimeter in size. A microscopy is required to view them. Their morphological differences enable one species to be differentiated from another. Schistosoma mansoni eggs on the left have prominent spines near the bottom which stick out at an angle, whereas Schistosoma hematobium eggs have smaller spines that stick out right at the bottom. Schistosoma japonicum eggs are more rounded, and the spines are hardly visible. We'll go over the morphology of these eggs again in the lab. Schistosomiasis is diagnosed by the detection and identification of eggs in fecal specimens for Schistosoma mansoni and japonica, and urine specimens for Schistosoma hematobium. Cases can be treated with anti-helminthics. To prevent getting infected, activities such as swimming or wading in fresh water in endemic areas should be avoided. Bath water should be heated to a rolling boil for 1 minute and cooled before use. Vigorous towel drying after accidental, very brief water exposure may help to prevent skin penetration by cercariae. Education and elimination of the snail intermediate host in endemic countries has helped to reduce incidence rates. Schistosome dermatitis or swimmer's itch occurs after swimming in contaminated water. There may be tingling, burning, and itching of skin within minutes of exposure, and small reddish pimples or blisters emerge within 12 hours. You can see some of the arm in the picture on the right. The rash is caused by an allergic reaction to schistosome cercariae when they burrow into skin. These are usually from birds. The rash will last for a week, then subsides gradually. Subsequent exposures, though, may result in increased intensity. Swimmer's itch is most prevalent during summertime in Ontario and other parts of Canada, especially in the Great Lakes region. It is not a reportable communicable disease, as it is not considered to be a public health risk and is not contagious since it cannot be spread from one person to another. Infected animals include ducks, geese, gulls, swans, and certain mammals such as muskrats and raccoons. Humans are accidental hosts. The life cycle of this type of schistosome cannot be completed in humans, and cercariae that penetrate skin do not develop further. Adult worms in the blood vessels of birds produce eggs which are passed into the environment through their feces. The ciliated larvae hatch in water to infect suitable snails that serve as intermediate hosts. Free-swimming cercariae penetrate the skin of birds and migrate to blood vessels, completing the life cycle. Swimmer's itch does not usually require medical attention. Corticosteroid creams or anti-itch lotions can be used to relieve itching. Alternatively, cold compresses or baking soda paste made by stirring water into baking soda until it reaches a paste-like consistency can be applied, or soaking in baths with colloidal oatmeal or Epsom salts. Scratching should be avoided to prevent skin infections, and in the case of severe itching, prescription-strength lotions or creams can be obtained by consulting with a healthcare provider. To prevent swimmer's itch, swimming or wading in areas where swimmer's itch is known or where warning signs have been posted should be avoided. This includes marshy areas where snails are commonly found. Toweling dry or showering immediately after leaving the water has been found to be helpful in preventing secarial penetration. Feeding and attracting birds in swimming areas should be discouraged, and health officials should be encouraged to post signs on shorelines where swimmer's itch is a current problem. When we look at the list of helminths identified in specimens that were submitted for ONP tests at the Ontario Public Health Labs, we can see that cestodes and trematodes are less frequently found than nematodes. Tapeworms were the most common cestodes identified.